Is it audible? Okay. Okay, let's start uh, today's lecture. Topic is the unit of cell, unit 3 of your syllabus. And in this unit 3, there are 3 topics mentioned there. One is cell is the basic unit of life, another one is the biomolecules, and third one is the cell division. So these 3 together form the unit 3, and 10 marks question are asked in your examination from this section. Actually 10 to 15 marks asked. You have to attend 10 marks. Options are there actually. So let's start here. So what is uh, life? So life means here we could say R cube. R cube means that is respiration, response to stimuli, and reproduction. These 3 together called the R cube. We could say if you go to Sundarban via a boat, and you will find a tiger there, just rest outside the mangrove forest in the sand bank. How can you know the tiger is live or dead? So you have to first of all make sound. If the tiger move back around or move its body, then you will come to know a conclusion that tiger is live. But still, if no response found, then you have to throw something, throw some cracker, or but still the tiger remains same in position, no response from tiger end. Then you have to go towards the tiger and check whether it respiration starts or not, respiration is going on or not. If respiration stops, there is no respiration taken by the tiger, then you will confirm that the tiger may be died. So response is a very important criteria for every life. One question asked in the NEET examination last 2 years back, that is an asked similar type question. There are several options are given, and the correct option for a life that is the response to stimuli. This is the primary option, and other things are the respiration definitely. Reproduction is one more thing, other also. So these are the basic rules of life. Now every life is made up of one cell or multiple cell. And what is the basic structure of the cell? There is plasma membrane. It's outside the membrane, one covering of the cell. That's the plasma membrane. One is the cytoplasm, and another one is the genetic material. Cytoplasm means everything, biochemistry, everything. The biomolecules are present. Physiological process is taken. That is inside the cytoplasm. And genetic material, that means the legacy passed from one generation to another generation. That is the parental cell to daughter cell. Every character is passed. That's why we get some parental character. You get from some parental character from your father side, mother side. So this is the genetic material, the characteristic feature from one generation to another generation, passed via some molecules. Those are maybe DNA, maybe RNA. So this basic 3 things are important for every cell: plasma membrane, cytoplasm, and genetic material. So this is the RBC, and this one is the squamous sick cell. You will both the cell you maybe find in microscope, normal simple microscope. In a school, they may show you this cell very easily from your cheek cell. They just take some smear of your cheek and with the help of some Giemsa stain or like some other stain, just a pinch of drop of stain is there, and you'll find this well slide, very good slide, and it be easily seen in any microscope. Here is how some interesting fact is that about 100 trillion cells are found in our body. 100 trillion cell. So can you imagine about that 100 trillion? After billion, there is million, millions, then billion, then trillion. So 100 trillion, very, very high. And in a tiny drop, look at a tiny drop here. This tiny drop here, and about 50 cells is present in this tiny drop. So imagine that one. This is I, there is a dot, and this is here is a dot. Is quite written in a different way. So that one is that. And here is the approximation of the cell size. Here, look here, from nanometer level to the hen's egg, big hen's egg. So about 65 nanometer. So this is the structure, virus structure, smallest one, but still present a coronavirus structure is a bigger one, about 300 to 400 nanometer size are found in coronavirus. So it is too large than HIV or bacteria. So it could be easily protected by normal mask also. So the other structures are here in our body. There's these two structures you notice here. One is the human red blood cell, RBC, and this one is the neuron cell in our body. This is the smallest cell in our body, human red blood cell, and this one is the largest cell in our body, that is the neuron, and largest also longest too, longest one also, longest cell in our body. So these are the basic idea about the cell, and this one is the human here. You look at as compared to the human here. Look here, this one. So very small, approximately the human hair. Just getting an idea how human hair length, and we have seen our hair just look like that, but under 200x microscopical power, you've seen this one, and this is here, this, and the bacteria, see here, this is the bacteria, here is the pollen grain. So imagine that the size, this is the bacteria, E. coli bacteria, and two E. coli bacteria if you join, that will turns into structure of the bacillus bacteria found in our curd milk. There is lactobacillus lactiti. So these are the structures, and this is the pattern-like structure. This is the pattern-like structure. This is called the RBC. Okay. And this is the lymphocyte cell. One lymphocyte cell means they are helping us to combat against the pathogenic attack. They perform the phagocytosis action, and RBC carry oxygen and carbon dioxide, and that's why they don't have nucleus for CO2 oxygen transport. Oxygen transport space is required much more, so nucleus is not there here. Next slide. Here, one scientist, the Robert Hooke, a very good scientist. Actually, he's an English scientist. In 1665, first discovered a microscope, simple microscope, that one simple microscope. And this is then the prototype of that. He made just one lens extra to the Galileo's telescope, and likewise magnification is done here, and he's seen some cork cell, like tiny boxes of honeycomb, tiny boxes of honeycomb, like honeycomb you see hexagonal cells are there, like that. So cork, what is cork? You will find in homeopathy medicine, old days homeopathy medicine, the glass vial, and there is the covered by a wooden structure cork. That one is the homeopathy. Present day homeopathy vials are plastic made, and you will not find the cork anymore. But in earlier days, homeopathy medicines are covered with cork. In some laboratory also, present day, that cork are also used. Present day rubber use. So cork is now, actually cork is a plant, sola plant. You'll find the Daket Saj in Durga Pratima. So they are being made up of that sola. So from that cork is harvested, and it is used for making the cover of some glasswares. So this is the cork, and Robert Hooke taken some amount of cork and make it sliced and seen under the microscope. So he found a conical structure and he designated these are the cell. And he also found the plant and in Pangia some their cells are present also. Here is another scientist called Leeuwenhoek. He also seen with a magnifying glass some animal clues in the pond water. Pond, if we go through the countryside, we will find green color water or brown color water. So took some water from there and see it in your magnifying glass or in your simple microscope you have. Then you will find animal cues or plant cues. Actually, the planktonic form you will find. These are the structures the Leeuwenhoek found in about 1674. Some books is there 1673, but I found it in 1674. Okay, next slide. After 150 to 100 years gap, now one question comes: how life comes in the present day Earth? How life comes? Is it spontaneously generated or something else, other planet it will come? So there is controversies are there. Evolution chapter we will discuss about these things, theory of creation, theory of God's creation, theory of pangenesis, everything. But one here is pertinent thing: life comes from is spontaneous generation. One point I'm asking you that is very interesting. You will find if you sometimes found any mouse, you could say mice, in your left cloths, or in a house you have long day back you come, you might be found some mouse, or you may find a maggot in a rotting meat. It is a common experience to us. So how mouse come? So Louis Pasteur is a scientist here. Louis Pasteur, you know, Pasteur Institute, a very big institute. Pasteur is also specialized in the biological field. If you found some mouse in your almirah, then you may question, if one question arises, how the mouse come in your almirah. Maybe if you go to cinema hall and took some snacks or some food like corn or like pea, and after some thing you taken some of the your lift up food is put it in your pocket and you forget it. So that is in your pocket and just hang the cloth in the almirah against again and close the almirah, or maybe forget to close the almirah and go away. After a few days you come back and you will find there is a mice. So the mice come from outside. How? Now it will found there is a food. For searching of food, it will come here and you will find. Sometime there are some ants may become. So is it spontaneous generation from your cloth? No, that not. That is coming from outside. So that's a simple thing I want to say. The life cannot be spontaneously generated. Rather, it could say life can form only from the pre-existing form. One another experiment. This is called beautiful experiment of Francisco Redi. Redi's experiment is called his scientist Francisco Redi. Here is the meat in a container, and here is meat in another container. Here is meat in another container. Maggots are there. Maggots are there present in here. Actually, outside the maggot fly come here and lay their egg, and you will find few days after there is maggots present. But if it is covered by a cloth and also some plastic paper or the foil, then no maggot will be found here after 2 to 3 days. So by this experiment, you could also say that life cannot become spontaneously within itself. One beautiful another experiment is the swan neck experiment by Pasteur. Here a broth media is boiled here, and this neck is a swan neck. It is the covered one. So dust or other microbes cannot enter this way because this is a bent part. So they cannot enter to this way. By normally it is quite difficult. So long time ago they are just come here, but the liquid present here is okay. So no growth of organism found in the broth. But if it is a straight neck one, then definitely there is some microbes present in the broth, and the broth is used by the microbes. So its control experiment is also done here parallelly. So to find out this one experiment. So it's a beautiful experiment. So we come to a conclusion here that life can come only from the outside, no spontaneously generated. Now we come here to scientists about the Sladen and Swan. These two scientists, German scientists, 1838, German scientist Matthias Sladen and one another scientist is Theodor Swan. So these two scientists here put their cell theory. According to them, every cell is every plant or animal body is made up of cell. And one another thing, here is the Rudolf Virchow. Rudolf Virchow is one German scientist, and he, according to him, every cell must arise from the pre-existing cell by dividing. So here is 2n cell, normal cell is dividing in another 2n by mitotic division. So from one cell to another cell, just division of the cell. So one cell can exist, one life can come from another life. So this one is the cell theory, another one. So these two together, we could say all organisms are composed of one or more cell. That means all organisms made up of the either one cell or more cell. One cell means bacterial cell, protozoal cell, one cell, and our cell, plant cells, these are the more cells, multicellular thing. So protozoa and bacteria are the unicellular, and rest are the multicellular. The cells is the basic unit, basic structure and function unit in all life. So this is two cell doctrine proposed by Sladen and Swan. And third one is the all cell are produced by the division of the pre-existing cell. So all the cells are dividing cell dividing property, and they are dividing themselves to give rise another two cell. So by this way, cell are multiplying. So we have seen it in the earlier slide here. These are dividing cell. So these three are together called the cell theory. Now we next to the slide. These four are another statement that each and every cell have DNA. As I have discussed earlier, that every cell passed it genetic information or characteristic features to the next cell by some hereditary information, by some hereditary molecule. We could say it would be DNA or RNA. So by this way, all contain hereditary information which is passed from one cell to the another cell during the cell division. Cell division, we have seen the chromosome is there. So by this way, they would pass. Within the chromosome, we know DNA is present. So all cells are basically the same in chemical composition. Every cell have similar type of composition, cellular components is there. DNA, protein, RNA, whatever may be. Other components is also there. And metabolic activity performed by every cell is the same. All chemical or physiological function carried out by the cell, physical movement, respiration, digestion, everything. Plant may perform food by the sunlight, using of the sunlight. Animal cell perform other different kinds of activities. Maybe it's secretory activity, maybe it metabolic activity, everything. So all the basic and chemical physiological functioning are carried out inside the cell. Phagocytosis is also physical activity. So all these are come inside the cell. Cell activity depends on the activities of the subcellular structure. Subcellular structure means mitochondria have different structure. We could say plastid have different structure, producing food. Ribosome, they are producing protein. Lysosome, they clean the cell. So by this way, every cellular component, subcellular structures are there. They have the different functions. So these four, along with the other earlier, these three cell doctrine together is a modern cell theory. Now we could say cell types, prokaryotic and eukaryotic. These the two basic type cells. Prokaryotic means those having the pro means earlier, karyon means the nucleus. So earlier type of nucleus is called the prokaryotic cell, and new type of nucleus is called the eukaryotic cell. Actually, prokaryotic cells are earlier one, and eukaryotic is the later developed one. How eukaryotic cell developed from the prokaryotic, that is a beautiful theory called endosymbiotic theory. We will discuss it in our next session. So you will find in your book this theory about this. I will discuss you, and if you have, I will discuss it in the next session. Okay. Now you will see here one prokaryotic cell. Here is the ribosome, protein machinery. Is the DNA. Here is the flagellum. Here is the capsule protecting the cell. So these are the structures of the bacterial cell. And prokaryotic cell, you know, its nucleus is not bound by membrane. Open nucleus is there. There is no membrane is there. So organelles are not bound by membrane also. Here is the paramecium. Here is the paramecium cordatum species. So this is the paramecium, and a nuclear bound by the membrane, and the process on many. Here the many organelles here, mitochondria, lysosome, these things are present within the cell. So this is the eukaryotic cell. So look here, the animal cell. This is the animal cell outside the protruding something there, the proteins and the carbohydrate lipid composition here. There's some vesicles transporting here through outside the cell. These are the internal structure of the cell. This is the nucleus. This is the tiny pore outside the nucleus called nuclear pore. From this pore, nuclear material come outside, and cytoplasmic material come inside to the nucleus. So this is the animal cell, and this one is the plant cell. You see here, similar like the animal cell. One basic difference is the animal cell and plant cell is outside the plant cell. There is a cell wall, but in animal cell there is no cell wall. So what is the difference between the cell wall, plant cell, animal cell? Fundamental two difference we could say. One thing, animal cell having no cell wall, but plant cell having a cell wall. And second difference is very important difference. There is the plant cell autotrophic in nature, but animal cells are not the autotrophic in nature. They are heterotrophic. That is, they dependent on some other cell. Although there are some exceptions like Eugloina. Now we could say, what is the function of the cell wall? Why is plant cell require the cell wall, but animal cell don't require of that? Actually, for this I will give you one example. There is, if you must say some football. Within the football there is a bladder, and it is covered by some other polypropylene material, polythene material, or synthetic material, or synthetic leather, like that structure is covered. So bladder is covered by the outside one another covering. So if you remove the other outer covering, and within the bladder if you put 30 psi, then the bladder will burst, definitely burst, because inside air pressure is too high. But if it is covered, then you may enter about 30 psi air, then it will not burst. So why it is not burst? Because there is a covering. So it has the power to protect the bladder from the internal pressure. So how it is similar to the plant cell? Because plant cell have the property to take water from outside, endosmosis. So plant take huge amount of water, and inside the plant cell there is a huge water pressure. We could say the osmotic pressure is here high. So high osmotic pressure can be mitigated by the cell wall, and that's why plant cell have the cell wall. This is the basic functioning. There are some other differences are there also. So I'm now going to there. So this is the end of the session. Okay, we will see you in the next session with plasma membrane and other cellular components. Okay, thank you.